Right, so next up, that you've already heard speaking, so next up we have Victoria Quiroga, and she will begin her presentation.
Okay, thank you, Kelly, so much to tell you our latest work. Hello, everyone. Today I'm going to tell you about our work entitled Ecological. Okay, are you seeing my presentation? Yes, good. Ecological
yes,
processes.
Sorry, we can't see you, but we can see your presentation.
Oh, okay. Ecological processes shaping the bacteria meta-community at different taxonomic resolutions. Okay, so as an introduction, microbes play a key role in Antarctic environments. And today you're going to meet Sierva Point Wetland Complex, which is a biodiversity hotspot in the Antarctic Peninsula. So as this is a wetland complex, in this work, we aim at determining whether the distinct environmental features would impose selection processes on bacteria community assembly, or this will be overridden by high hydrological connectivity during summer, which will favor high levels of dispersal, imposing biotic homogenization. So also, if these assembly processes would operate in the same way at different taxonomic resolutions, in particular, OTUs and ASBs. Okay, my camera is on, so I hope you all can see me. I only can see my screen only. So this is Sierva Point. It's an Antarctic specially protected area of around 1 square kilometer. The sampling was performed during summer 2018. We took 64 samples from 5 different habitat types. Bacteria community was studied with high throughput sequencing of 16S rRNA gene V3B4 region with Illumina MySec. And as you can see in the picture, the research was conducted at Primavera Station, which is managed by Argentina. Okay, so almost 20 years ago, Agras and collaborators classified Sierva Point Wetland Complex into environmental units. You're going to see the environmental units on this map as numbers. This classification was based on topography, land cover, and vegetation type. And you're going to hear me many times during the talk referring to environmental units. So you can see the map to take a picture of how they are distributed at Sierva Point. Well, we apply the approach proposed by Stegen and collaborators to study assembly processes at these 2 taxonomic resolutions. This approach proposed 2 indexes, beta nearest taxon index and Bray-Curtis-based raw creek. These indexes quantify the degree to which phylogenetic and taxonomic turnover deviates from the null model expectation. And based on the values of these 2 indexes, we can estimate the relative importance of each assembly process. Homogeneous selection, variable selection, homogenizing dispersal, and dispersal limitation. I will refer to undominated, where neither selection nor dispersal was the dominant process. Well, I told you we sampled 5 different habitat types. So here you have pictures where you can see them: mosses, soil, snow, water, and epithelium. So for environmental data, we have pH, conductivity, slope, penguin presence, habitat type, and the environmental unit in which the sample was located. So we put it all together in a PCA with mixed data. And as you can see, some environmental differences are observed between habitat types. Well, We analyze the sequences with data 2 to obtain ASV cluster into 90% similar, data 2 to obtain ASV cluster, ASV table. We clustered ASV into 97 similarity to obtain the auto table. And regarding OTUs, more than half of it was shared among habitats. And as you can see from the PCOA ordination plot, no clear phylogenetic differences were observed between habitats. So looking at the assembly processes, bacteria metacommunity structure based on OTUs revealed a mainly stochastic system undominated by any particular process. On the contrary, when we look at ASVs, most of ASVs were habitat specific. And as you can see at the PCOA ordination plot, phylogenetic differences between habitats were observed, kind of resembling environmental patterns. So, assessing for assembly processes, okay, a completely shift. Here, bacteria metacommunity structure based on ASVs was mainly determined by homogeneous selection. The higher taxonomic resolution of ASVs and BL deterministic processes probably related to microdiversity patterns not detected at the OTU level. Okay, so the former comparison, you can also visualize it through beta NTI histograms. Now you're going to see for OTUs and for ASVs. And for ASV, you're going to see that histogram values move to more negative ones, resembling the strong homogeneous selection both within and between habitats. So this lower observed ASV phylogenetic turnover as compared to the new models are likely to be due to recent evolutionary divergence between close relatives that exist in different habitats. Okay, this is a diagram for you to check how we look for variables related with selection and dispersal. First, we combined environmental variables with spatial eigenvectors in a mixed PCA. The resulting PCA mixed dimensions or axes were used as explanatory variables in a distance-based model selection procedure with beta nearest taxon index or Bray-Curtis-based Row-Crick distance matrices as response variables independently. So finally, for each of the PCA mixed dimensions selected, we check the variables loading on those axes. So we did this model selection for ASV metacommunity structure, and we found that habitat and pH impose homogeneous selection at a local scale, while environmental unit appears to impose homogeneous selection probably at a landscape level. Remarkably, eutrophication by penguin guano also imposes homogeneous selection. Before talking about dispersal, I would like you to look at these two Google Earth images. I put them just for you to realize how Tierva Point gets completely covered by snow during most parts of the year, from April to December. And we believed that most of the dispersal happens only during the snow-free summer month period, and that the snow cover is heavily restricting dispersal during most parts of the year at both local habitat and landscape environmental unit scales. So based on our results, we propose this hypothesis that macroevolution occurring within microhabitats during the snow cover low dispersal period of the year is a primary factor governing the phylogenetic composition of the bacteria metacommunity across Tierva Point wetland complex. So as you can see from the model, dispersal is heavily restricting during most parts of the year, which is snow cover, Dispersal only happens during summer months, and selection evolution appears to be occurring in kind of isolated microhabitats in this wetland. So probably this is a new pattern for bacterial assembly, but we believe that maybe if other ecosystems have this heavily restricted dispersal during most part of the year, maybe we can find some similarities in assembly processes. So I think I talked too fast. That's all. I want to thank you all. Also, all my co-authors. Without them, this work couldn't be possible. I'm also at the poster, so at the poster chat, you can ask me wherever you want. And I would like to especially thank Kylie and all the ISME Unity and Diversity Organizing Committee for this opportunity, which is completely wonderful.
All right, thank you so much. Thank you, Victoria. All right, so we have time. Do you see me? So I can't see you. No, I can't see you. No? But we've been able to see your presentation just fine. I just can't see your face. I'm not sure.
See if you can see me now?
No. Do you have something covering your camera?
No. But I think I moved this thing, the webcam.
Oh, okay. It looks like.
Sure.
You know, it looks like everyone else can see you, just not me. So.
Okay.
That's all right. Okay. All right. All right. So I have one question. What is your reasoning for using, or I guess analyzing, both at the ASV and OTU level, or I guess methods?
Okay. Well, the second approach was firstly developed for OTUs, but I think we are seeing so many papers talking about ASVs like ecotypes or strains. So I think in some ecosystems, I think it will be most common to see that OTUs and ASV follow the same pattern. But here, I think this is a special ecosystem because although you can see this is too green, it's a wetland with different habitats, but it is located within Antarctica. So it's quite isolated from the rest of the world. And as we didn't find differences between OTUs, so we tried to deeper dig into the ASVs to see if we can see some microdiversity patterns that, well, were there, but with OTUs, we couldn't see them.
All right, well, thank you very much. Yes, there were several more questions, but we don't have time, so please, audience members, please definitely reach out and hopefully you can get your questions answered. And thank you again.
Thank you.
All right, so now moving on. All right, great. All right, so next up, we have Mafalda Baptista. And sorry, we cannot hear or see you. Okay, now we can hear you. I cannot see your face. Oh, your screen was sharing. Yes.
My screen is sharing?
Your screen looks good. I think you need to turn your camera on.
I think it is on.
Oh, yes. Okay,
and the screen is sharing?
Yes, it is sharing. and you can put it in presentation mode.
Is it good now?
Yes.
Okay.
Well, actually, I can actually, I think you need to move it to a different screen. I can see your second slide.
Okay. Not sure if I can do that. Am I still sharing my screen?
No, not right now.
Okay.
Okay, so now you are sharing.
Is it sharing?
Yes, it is sharing. I can see your computer.
Let's just try to make a presentation view. Is it possible like this?
Yep, no, it looks great.
It looks great?
Yes.
Well, thank you so much, Kaylee, and good evening, everyone. I will be talking a little bit about the work that we have been doing as a collaboration between the University of Porto in Portugal and the University of Waikato in New Zealand through the Portuguese Polar Program, Antarctica New Zealand, and also the Korean Polar Research Institute, which have been our partners on the field. And I will be talking about the biogeography of microbial communities around the penguin colony in Antarctica. And I will start by talking a little about Antarctica soils, which are generally considered one of the most oligotrophic soils in the world, with the exception of penguin colonies. Due to the guano that the penguins produce, they can be a source of nitrogen input into the ecosystem that can potentially be spread through the inland terrestrial oligotrophic ecosystems. So we use this as a case study to see how nitrogen is transferred, to track nitrogen source and nitrogen circulation around these habitats. So we did this at the Adair Peninsula in northern Victoria Land. You can see it here on the Antarctica map. And you also can see here that the Adair Peninsula is a landmass that spreads into the Ross Sea. And at the very top, you have Cape Adair. And next to it, at the sea level, you have a triangular shape that is Ridley Beach, and that currently harbors the biggest Adelie penguin that we have on record for the Antarctica continent. You can see it much better here on this picture, a photo taken from the top of Cape Adair next to the automatic weather station, in which you can see over here at the beach the penguin breeding ground. Cape Adair is also very important in the history of Antarctica exploration. It is the site where the first expedition spent the whole winter in Antarctica at the turn of the 20th century. And you still can see nowadays the huts that the Southern Cross expedition built back then. And for that reason, it is also an Antarctica specially protected area for its historical interest. At this site, you can see that the types of soil that we could collect are very different. The ones that you see on the beach are mostly shingle-like soils with a lot of glacial debris. And up on Cape Adair, you could see that the soils are much more rockier and with a coarser grain. The better part of these soils are covered in ice or are covered in snow throughout the better part of the year. And we have sampled in these different systems to check for the soil microbial community. And we did that by analyzing the V4 region of the 16S ribosomal RNA gene. And we have also determined soil abiotic factors that could be of interest. And I will start by talking a little about those soil abiotic factors. So you can see here, you still can see my presentation? Kaylee?
Yes, but it's no longer in presentation mode.
It is not?
It is not in presentation mode, no.
Let's just give it a little bit of time, perhaps. Is it coming back?
I can just see your computer. Can you?
I was changing the slide and it went into.
Oh, no.
I'm so sorry for this. It doesn't seem a very good system to work with two screens at the same time. How's it going now?
I can't see anything. I don't think you're sharing. Okay, now I can see your computer. Yeah, so now I can see your computer.
What about now?
No.
Oh, this is disappointing.
I'm so sorry.
Oh no, I am apologize.
Okay.
I don't know what's going on.
Okay, it might also be my side of it. Okay, I can see you. I can see it now.
You can see it. It's on presentation mode.
Yes.
Okay. So what you can see here is I would like to go back one slide. No, it's not working. Oh. What you can see here is the sampling effort that we undertook at the Adair Peninsula, covering Ridley Beach and the adjacent Cape Adair. And I will just focus on the results that we got for ammonium and nitrate. You can see that they have much higher concentrations at the beach and at the tip of Cape Adair, where you also see the penguins are breeding. And what you also can see on the spatial distribution maps of these nitrogen forms is that they are not scattering throughout the rest of Cape Adair. So we inquired what would be the reason that was stopping these nitrogens form to being spread throughout this ecosystem. And in fact, when we checked the data from the automatic weather station, we could see that the prevailing winds were coming from the south throughout the Adair Peninsula. So in fact, the guano that was being deposited at the tip of Cape Adair was being blown into the sea and was not contributing to be spread throughout the Cape Adair and the Adair Peninsula because of that. We further looked into how the carbon and nitrogen content was spread out throughout this ecosystem, and we focused on a transect encompassing penguin samples that were collected on the beach, one penguin sample that was one penguin breeding site sample that was collected at the top of Cape Adair, and other samples at the top of Cape Adair that we did not see any presence of penguin in. And so what you can see here is that similar to the results we had for the nitrogen forms, the content of carbon and nitrogen decreased as we went further away from the colony. But in fact, when we look at the ratio between carbon and nitrogen, we can see that for the breeding site at the top of Cape Adair, where we see penguins, we do have a signature of penguin presence because that ratio between carbon and nitrogen is much lower than from any other sampling sites, which indicates that the penguins are inputting an extra nitrogen source, but not a carbon source to compensate for that excess. We further looked into nitrogen sources by analyzing the stable isotopes 15N and 13C. And what you can see here is that the samples that we got from penguin breeding sites were much closer in their 15N and 13C content to penguin guano than the samples that were collected in mineral soils where we could not detect any penguin presence, indicating that this penguin guano 15N was increasing the content of nitrogen in these soils. And we have further looked into the way that nitrogen was being circulated into this ecosystem by looking into ammonia 15 oxidation rates as a potential measurement of nitrification into the ecosystem. So in Antarctica soils, nitrification rates are usually very low, but our results show that places where penguins breed, independently of being on the beach or on Cape Adare, showed a much higher potential nitrification rate than sites where penguins were not present at all. So we further looked into this process of nitrification by encompassing the results that we had gathered from our 16S analysis. And specifically, we went looking for archaeal and bacterial populations that were involved in the nitrification pathway of the nitrogen cycle. And here I show you the results we got for the relative abundance of these organisms. So you can see there is a sharp difference between archaeal and bacterial populations that are involved in the first step of nitrification. As we would expect, archaeal are much more present on sites where we have lower concentrations of ammonia. And you can also see that the bacterial populations were mostly present on sites where we could see penguin breeding. When we analyzed bacterial population that were involved in the second step of nitrification, the knob bacteria, we found out that we had a missing link for this ecosystem, as we could only report their relative abundances in places where we could not see any penguin breeding. So to further look into the way that nitrate and ammonia could be driving these processes, we performed a correlation between this relative abundance of specific ASVs that we had found and the concentrations of nitrate and ammonia. And you can see that they were positive, slightly higher in the case of ammonia, but basically selecting for the same two ASVs throughout the ecosystem, which for us was a very interesting result, as we had seen that the archaeal community was much more abundant, and we could not get any strict correlations between their presence and the content in ammonia. So to conclude this presentation, I would just like to say that with this work, we have seen that the penguin colony at Cape Adare could transfer nitrogen from the near-shore environments where they breed into the inland oligotrophic terrestrial ecosystems of Antarctica, and that this gradient of N availability that we could see around the colony actually drove differences in the type and the amounts of nitrogen forms that were present that had implications on the nitrogen recycling processes and on the communities that are involved in these steps. So our aim in the future is to further explore these archaeal and bacterial populations to characterize them in terms of taxonomy and in terms of their metabolic processes. Thank you very much for your time, and I am sorry for all the problems that we had with this presentation.
No, thank you. No, please don't apologize. No, it was great and not your fault at all. We're all kind of
I'm so sorry.
Together. No, please don't apologize. No. All right, so we have time for one question. All right, I'm not seeing any questions. I'll just give a few more seconds and then we will move on. But again, thank you so much.
Thank you, Kaylee.
I was wondering, I love penguins. I don't have any intelligent questions to ask, but it was
so good. No worries, it's so good.
All right, so we will move on. But yeah, so thank you again. And if you do have any questions that come to you later, attendees, please reach out to Mafalda and ask your questions. All right. All right, so next up, we have Songlin Chua, and they will be giving their presentation. Can you, let's see. Can you hear me? Oh, perfect. Okay, all right. So I don't, let's see. All right, right now, okay. Oh, perfect.
Yeah, okay. Okay, so can you see me too? And my screen?
Yeah. So I personally can't see you. I think this might just be a program, but I do see your webcam on, so I think at least some people can see you.
I guess so, yeah.
Sorry, but I could definitely hear you and I could see your presentation.
Okay, then that's good. Okay, sure. Okay, so probably I will start my presentation then.
Yes, you're
Okay,
good to
so yeah. Okay, so thanks once again, Kaylee, for your invitation, as well as ISME for organizing this summit itself. Okay, so in any case, right, today my talk will be about engineering trap and then release mechanism. Okay, whereby the biofilms are able to remove the microplastics from the environment itself. Okay, so in this case, right, just a bit of introduction, okay, for all of you. I think all of you here should be very familiar about microplastics pollution, or rather the plastics pollution itself. But for microplastics, right, they're actually a very small piece of plastic itself, and they are usually less than 5 mm in diameter, okay. And because they are very small, right, they have a huge impact on the food chain itself. So for example, right, in the primary microplastics, where it can come from microbeads from your cosmetics, your toothpaste, and of course they can come from your clothes, things like that. They are actually very small particles, okay, which can easily enter into the wastewater and of course into the natural environment. And of course, when we have a degradation of larger plastic things like, for example, plastic bags, plastic bottles, okay, into very small microplastic particles that can also enter via rubbish, okay, or certain kinds of leaching into the rivers or lakes. So in this case, microplastics have been found in a lot of diversity as well as life in the oceans, okay. And in fact, this actually results in the transfer of microplastics down the food chain. Eventually, it will definitely reach our dining tables on its own. So in this case, right, microplastic is a huge problem, okay, all across the world. And because they are very small, it's really difficult to be removed, okay, from the oceans or other water bodies. And the prevalence of the microplastics is quite a serious problem because, in fact, there are many studies done across the world globally, and this is just one example in the Pacific Ocean, whereby they have already quantified the number of microplastics, okay, in the ocean, and they found that there's really quite a lot of particles, okay, nearing thousands of them, especially if you are much closer towards the cities or the land itself. Okay, so that also means that the spread of and the prevalence of the microplastic is really a huge problem in terms of removal later. So usually if you have to remove them physically, it's usually larger plastic particles, but for those very small particles, it's really difficult to remove them. And in fact, right, it's even more difficult to remove them from using current technologies, okay, especially in the wastewater treatment plants. So in the wastewater treatment plants, we try to remove them using filtration, but in fact, it's actually not very effective in removing them. So in fact, some of those studies have shown that the efficiency of removing microplastic can range around 70% to 90% of the microplastics, okay, from the wastewater. So that also means that maybe 10 to 30%, okay, of the microplastics can remain within the effluent and therefore be released into the environment. So this actually implied that a lot of microplastics can pollute, okay, the environment, especially where it's very close to the wastewater treatment plant. Okay, and in fact, in Hong Kong, where we are studying over here, we actually found that there's quite a lot of microplastic, a higher concentration of microplastics within those areas where there's more human activity, or it actually comes from the leakage of the wastewater treatment plants. So there are actually a couple of studies done in recent years about the microbial plastisphere. Okay, so they are just basically microbial ecologies where they are able to live in the microplastics itself. So basically, microplastics are very small, and therefore people have already studied and found quite a lot of microbes that are able to colonize as well as survive on those plastic environments. So there are many different kinds of plastic, like for example, PETs, PVCs, and things like that. So in this case, right, people have also found that bacteria can grow slightly differently, okay, on different kinds of plastics. And for example, you can have diatoms, you can have bacterial fungi growing on top of them. So in this case, right, if you study the microbial communities from different places, okay, from different that are actually isolated from different places, like for example near the wastewater treatment plant, you can find more of the E. coli, or rather human related kind of bacteria, E. coli, Pseudomonas aeruginosa, or some, and of course there's also other environmental bacteria like Pseudomonas putida colonizing on those microplastics themselves. Okay, so people have been trying to identify ways to remove microplastics. Okay, and in fact biological methods are being considered. So in fact, right, there's enzymes like for example PETs, which have been identified to degrade PET. But of course, the degradation of the microplastics and other plastics are probably not a very good idea, because there's definitely release of other toxic compounds, which is why we're actually thinking of whether is it possible to bioaccumulate those kind of microplastics, okay, for example, in a bioreactor or in wastewater treatment plants, so that we're able to recover all those microplastics, either for incineration later or for even recycling later. Okay, so in fact, for proof of concepts, we actually use environmentally prevalent bacteria, Pseudomonas aeruginosa. We also try other bacteria, but in any case, right, those kind of bacteria, they are very good at forming bacterial biofilms. They are able to aggregate as well as trap microplastics themselves. So in this case, right, as an example, we use a black PVC microplastic. Okay, so that's around 100 microns in size. So in this case, right, we are able to observe that Pseudomonas aeruginosa is able to create a kind of a bolus of microplastic. They actually aggregate them together, okay, and therefore this allows the possibility that whether this actually raises the possibility of whether we are able to aggregate all those microplastics and trap them for convenient recovery later. So in this case, we are able to show that over time, the amount of microplastics, the PVC that we are using in this case, are able to be trapped at a higher rates. So that also means that probably we are able to either genetically engineer those kind of bacteria, or we are able to identify certain environmental isolates which are able to trap those microplastics at a much higher rate itself. Okay, so we also tried to check whether this kind of bacteria are able to trap different kinds of microplastics, whether they have different size, material, or even density itself. So in fact, right, when we actually use, for example, PET, PMMA, nylon, or even polystyrene, which is able to float, okay, because their density is higher, we actually found that Pseudomonas aeruginosa are able to form biofilms very efficiently on them, okay. But of course, they are still not very good at forming biofilms. That's why we are trying to identify better ways of trapping them, okay. So in any case, right, I will try to confirm whether those kind of material done by Pseudomonas aeruginosa are really biofilms or not. So in this case, right, we did a bit of imaging, and we found that those kind of bacteria actually colonize quite a lot on the microplastics themselves. So we did a bit of confocal imaging, and we tagged the bacteria with GFP, and we found that okay, there's really bacteria colonizing a lot on the microplastics. And in fact, when you take a close look using the SEM imaging, you can also identify quite a number of bacteria colonizing on top of it. And of course, the formation of the bacterial biofilms are definitely done by cyclic diGMP signaling. I think most of you should be kind of familiar with bacterial signaling in this case. And the cyclic diGMP signaling itself is very important for biofilm formation. So the increase in cyclic diGMP levels is usually correlated with biofilm formation, and the decrease in cyclic diGMP signaling will definitely be correlated with biofilm disposal. So in this case, right, the synthesis of cyclic diGMP is mediated by the diguanate cyclase, the DGC itself, and of course for the degradation of cyclic diGMP, this is mediated by phosphodiesterases, which is the PDE. So we were wondering whether is it possible to manipulate this cyclic diGMP signaling so that we are able to either boost the biofilm formation, and then after that we are able to degrade the biofilm for convenient recovery of the microplastics. So in this case, right, we try to first show that the biofilm itself is really controlled by cyclic diGMP signaling. So we actually try to measure the cyclic diGMP concentration, okay, and we found that indeed there's increased amount of cyclic diGMP, and there's also increased production of the exopolysaccharide concentration. So in this case, right, when we use a biosensor, which is a CDI-GFP biosensor, so in this case, right, this CDI-GFP correlates directly to the upregulation of cyclic diGMP signaling. And we actually found that there's increased production of the GFP expression. So that also means that this is really indeed a biofilm, and therefore we are probably can identify ways to promote biofilm formation over here. So in this case, right, the loss of exopolysaccharides, okay, from the biofilm actually eliminates the biofilm's ability to trap the microplastics. So that also means that by manipulating the cyclic diGMP signaling pathway, probably we are able to boost the exopolysaccharide production in this case. So we did a bit of screening of the DGC mutants. So we have a bit of our in-house library, and we tried to identify which kind of cyclic diGMP signaling gene is actually very important for trapping of the microplastic. And in fact, we actually found a couple of them, but one of them, which is the most important one, will be the WSPR. So when we actually use the WSPR mutants and try to test its ability to trap microplastic, we actually found that there's actually decreased ability, okay, up to 3 times as compared to the wild type itself. So that also means that the WSPR is not very efficient at trapping of the microplastic. So this actually raised the question of if we are able to upregulate the WSPR operon, okay, so that it's able to increase the trapping of the microplastic. So in this case, right, we try to overexpress the WSP operon, okay. So if, for example, if you take a look at the figure on the left-hand side, so there's actually a regulator that's regulating the WSPR DGC on its own. So under normal circumstances, the WSPF is able to downregulate the WSPR, okay. So if we are able to use a WSPF mutant, okay, so this actually de-inhibit the expression of WSPR, resulting in the increased expression of the exopolysaccharides, and of course this will boost the biofilm formation. So in this case, right, where we use the WSPF mutants, right, we actually found that this mutant is really able to form a lot of biofilms on the microplastics, and this increases the efficiency of trapping of microplastics in this case. In fact, when we increase the amount of microplastics concentration within a bioreactor, the WSPF is able to even aggregate much higher levels of microplastics as compared to the wild type Pseudomonas aeruginosa itself. So when we look under the SEM itself, we also saw that there's really much more of the bacteria colonizing on top of it. And because it has increased biomass, this actually causes all of the plastics, okay, the microplastics to actually sink to the bottom of the bioreactor. So if they are able to sink to the bottom of the bioreactor, that also means that we are able to remove, recover, and retrieve all those microplastics that's being trapped within the biofilms very easily. So this actually same goes to those lighter microplastics, which are very difficult to remove. Like for example, your polystyrene, because they are very light, they are able to float on water itself. Therefore, once they sink to the bottom, right, it's very easy for us to recover them. Okay, so we also test their versatility of these mutants, whether they are able to grow at different temperatures. So basically, right, we try different temperatures and different kinds of water, whether is it in fresh water or salt water. Okay, so in this case, right, by adding a bit of carbon source, okay, we are able to see that this kind of mutant is able to trap the microplastics at much higher efficiency as compared to the wild type itself. Like for example, when we use 30 degrees, 25 degrees, and of course we also use 16 degrees. But of course, at 16 degrees, it grows a bit slower as compared to 25 degrees. So in this case, right, they are still able to trap quite a lot of microplastics as compared to the wild type itself. So by trapping the microplastics, right, we are able to easily recover all those microplastics. But of course, if you want to use them for example, recycling, or maybe you use them for combustion, incineration later, right? Of course, we have to release them from the biofilms themselves, okay, because the biofilms are really sticky. So how are we able to engineer something what we call by a release mechanism, okay, to allow the biofilms to release all those microplastic by themselves, okay, without any external interference, okay. So in this case, right, we try to introduce another gene, okay, which is a release mechanism into the bacterial biofilms. So in this case, if you take a look at the left-hand side figure, so in this case, when you insert a plasmid with a release function, this actually contains a PBAT promoter. So this is actually arabinose inducible. Okay, I mean you can insert any kinds of stimulus within it. Okay, this is just only a proof of concept over here. So in this case, right, there will be a YHJH PDE gene. Okay, so this PDE gene is able to cause the reduction of cyclic di-GMP and therefore results in the disposal of the biofilm. So we actually found that for the usual WSPF on its own, when you add in the arabinose, which is the inducer, we are unable to cause the disposal of the bacterial biofilms. But when we actually used the WSPF PBAT YHJH with the release function, you can see that with increased addition of arabinose, you are able to increase the release of the microplastics from the bacterial biofilms. So this actually causes the breakdown of the bacterial biofilms, and eventually this releases all of the microplastics back into the media. So that also means that we are able to trap the microplastics in the first place, okay, and then we retrieve them so that we are able to cause the biofilms to disperse and release the microplastics later in another bioreactor. So this is a very convenient kind of recovery where we are able to do that. So as a proof of concept later, we actually try to collect some of those environmental samples. Okay, so we collect the seawater samples from the vicinity of a sewage outfall, where microplastic pollution is still very prevalent. Okay, so this is actually done within Hong Kong itself, and we know that there's still a very poor kind of trapping efficiency of microplastics itself. So in this case, right, we actually found that this kind of bacteria in the bioreactor are can very easily accumulate microplastics, okay, from those samples, and it's actually not very it's actually quite indiscriminate in trapping different kinds of microplastics. So in this case, right, for example, you have PEHD, PET, PP, polystyrene, and polyethylene, propylene. So it's able to trap really different kinds of microplastics, and therefore after that, right, we are able to release them using the release mechanism. So the amount of microplastics that remain within the bacterial biofilms, okay, after the release mechanism is very little, and we are able to retrieve the rest of most of them after the release mechanism. So this actually implies that our engineered shrink is able to do that very efficiently by trapping the microplastics, and then subsequently we are able to release them in another bioreactor. Okay, so what actually really means for the future? Okay, so in this case, right, we are just only having a bit of a proof of concept over here, and in fact currently we are trying to identify and select the natural, okay, kind of probiofilm microbial species in the plastic sphere itself. So basically we have identified Pseudomonas putida, and we are currently working more on it. Okay, and of course we also did a bit of engineering on them, and we found that Pseudomonas putida itself are really also able to trap those microplastics. Okay, and of course for the release mechanism, we also identified a couple of anti-biofilm reagents. Okay, so like for example, nitric oxide have been previously shown to be able to disrupt to cause biofilm disposal. Okay, they can also so some of those other enzymes probably they can disrupt biofilm matrix, and therefore this can also promote the release of the microplastics from the environment. Okay, so basically, right, addition of the disposal agent can cause the bacteria to disperse, or we can directly break down the entire biofilm matrix using the biofilm disrupting agent. So this actually helps, okay, in the release of the microplastics, so that we are able to conveniently recover them later. Okay. So in summary, my presentation is just basically the proof of concept whereby we are able to induce the biofilm formation around the microplastics, okay, so that we're able to accumulate all of them, okay, into a larger mass, so that we can easily filter them out, or because they are heavier, they actually sink to the bottom of the sedimentation tank, so that we're able to conveniently retrieve them later. So in this case, right, after that, if you want to recover them for either incineration, or if you want to use them for recycling, we are able to cause the biofilm disposal using some kind of stimulus, and therefore this releases the bacterial biofilms, and this releases the entire microplastic setup itself. So this is, I think, a very interesting kind of a phenomenon. We are able to retrieve those kind of microplastics pollution itself. Okay, so in this case, right, thank you very much, and I would like to have special thanks to my institute, Hong Kong Poly U, as well as my department, as well as all of my collaborators from other universities. So thank you very much for your attention. I will be happy to take any questions.
All right, thank you very much. So we unfortunately don't have time for any questions, so please send a private message or reach out independently to ask any questions. There are several in the chat box. All right, thank you. So now we're going to move on. Thank you so much. Thank you. Thank you. All right, now we're going to move on to, we have Miao Xiao Wang, and unfortunately, they are currently in Beijing, where it is nearly 6 o'clock in the morning. So instead, I will share a presentation they recorded ahead of time.
Hello, everybody. I'm Miao Xiao Wang. You can also refer to me Roy. I'm a PhD candidate of Peking University, China. It is my great pleasure to present my recent work in this great meeting. In this talk, I will share a story on how to stabilize metabolic division of labor in microbial communities and how these communities assembled. In human society, when we try to perform a complicated sequential task, for example, produce an expensive car, we may adopt two strategies. First, we can find a Superman to carry all of the working steps, but usually the Superman is busy and tired. Alternatively, we can find several normal workers, and each worker is specialized in one specific step, so they can finish this task by division of labor, which is thought to be a more smart strategy. This kind of trade-off does not only happen in human society. In microbial world, bacteria also work hard to transform an initial substrate to the final product with several intermediates and several reaction steps. In addition to evolve to the superbug, bacteria are also very smart to distribute the steps among different populations, forming an interesting kind of social interaction called metabolic division of labor. Metabolic division of labor is frequently observed in microbial world. A classical example is nitrification, in which the first bacteria transform ammonia to nitrate, and another bacteria convert nitrite to nitrate. In engineered systems, people also apply this strategy to synthesize valuable product. Although this is an attractive strategy, how the MDLL system self-assembled remains poorly understood. In other words, we need to know whether the members in these systems are happily to work together. To address this question, I chose naphthalene degradation pathway as a representative system and segregated the long pathway into 2 or 4 steps. Thus, I constructed 3 synthetic microbial consortium engaged in 2-step MDLL and 1 more complex consortium engaged in 4-step MDLL. Naphthalene degradation pathway represents a variety of pathways of Hendry carbon degradation. A very important feature of these systems are the final products here, those organic acid or CoA, are super tasty food for bacteria. In contrast, the intermediates are always associated with complicated chemical structures, which cannot be directly used as carbon source for growth. Thus, if we distributed these pathways into several steps, the guy perform the last steps should be super happy because they can first get the food, while the population perform the former steps will be super sad because they pay more but get less. This unfair product allocation may destabilize the MDLL system. Consistent with this prediction, when we cultured our synthetic consortia using naphthalene as the solo carbon source, all the consortia grew poorly, in which the population performing the last step rapidly exploded and largely dominated the consortia. When we diluted these mixed cultures and passaging them into a new fresh medium, the growth of this consortia becomes even worse, suggesting that these communities were ecologically unstable. This phenomena is just like the lost worker in the assembly line steal the final product and do not share money with the former workers. So we named the population performing the last step as embedder and named the instability caused by embedder as the embedder dilemma. Therefore, weakening the asymmetric benefit of the embedder is key to maintain stability of the MDOL system. To figure out a rule guiding our design of stable MDOL system, we then constructed a mathematical model to describe dynamics of MDOL system. We first simply focused on a two-step MDOL system. We named the first population as 1, 0 and the second population as 0, 1, and this 0, 1 behaved as the embedder. And also, in this model, we include multiple factors, including the reaction speed of each step, the transport speed of intermediates and products, and also the metabolic burden of the two steps. Also, we hypothesize the end product is the solo carbon source for the bacterial growth, thus consistent with our hypothesis of embedder. From observation, we obtained a simple formula which points out that the final frequency of 0, 1 is determined by the quotient of n and m, where n reflects the provide benefit of 0, 1, and m reflects the growth advantage of 1, 0. This formula also suggests a condition to maintain the coexistence of the two populations. That is, the 1, 0 should hold a growth advantage at 0, 1, and this growth advantage must higher than the provide benefit of 0, 1. This mathematical rule can be visualized by a density map. As shown here, the two populations coexistence only when n is less than m, and the steady-state frequency of embedder can be directly calculated from n dividing m. In contrast, when n over m, the community is unstable, where the 0, 1 rapidly exploded and excluded 1, 0 at the end. That is, the embedder dilemma happened. Then, we modified our basic model to consider some additional mechanisms. For example, if intermediate or product can be lost via abiotic conversion, the simulations show that the parameter space of coexistence become narrow, which indicates that the community becomes more difficult to be stable. In another case, if the substrate is toxic, the basic formula is modified, which suggests that substrate toxicity favors the growth of 1, 0, and thus the two populations becomes easier to be coexistence. After the mathematical analysis, we turned to perform experimental tests using our synthetic microbial consortium. Here, we set the experiments with the first consortium as an example. To generate mathematical predictions, we added specific pathway mechanisms to our basic model. For example, the toxic effects of naphthalene as well as the intermediates 1,2-hydroxynaphthalene were experimentally measured and included in the model. We then determined the M value of this system is 2.53, so derived the prediction of the community assembly. Subsequently, we aimed to increase the value of M to obtain stable communities. To modify M values, we constructed 3 soothing modules, where the expressions of toxic protein CCDB or X175 were controlled by an inducer Ramolase. We delivered alternative module into cells of embedder cells to modify their growth rate. The quantitative fitting analysis links the death rate of embedder to Ramolase concentrations. Since M is directly associated with D2, the experimentally modified of M can be realized by setting up randomized concentrations. We next co-cultured the modified embedded strings with the original 1,0 strings, and the results show that when M was located in a suitable range, the community exhibit stable dynamics. In these cases, our mathematical rule will predict the assembly of this consortium. Next, expanded our mathematical model to predict the assembly of the community engaged in multi-step MDLL. As the previous model, we include multiple factors, including the reaction speed of each step, the transport rate of different intermediates and end products, as well as the metabolic burden of each step. Through numerous mathematical simulations, we developed a more general formula to define the final frequency of the embedded population in an n-step MDLL system, where NK reflects the relative benefit of the embedder against the case population, while MK reflects the relative growth advantage of the case population against the embedder population. Taking three-step MDLL as an example, simulations revealed that the coexistence of the three populations is only present when the value of NK and MK is belonging to the range defined by the formula. This formula can also be applied to explain the results of the mathematical simulations with MDLL systems, including more steps, suggest the generality of this rule. To experimentally test the rule, we cultured our synthetic consortium engaged in four-step MDLL. We applied the similar strategy as before to experimentally modify the death rate of the embedded string, and thus modify the value of MK. In these co-culture experiments, we observed the stable community dynamics when the values of NK and MK are in a suitable range, which is consistent with our mathematical prediction. In summary, we built a theoretic framework to predict the stability and assembly of microbial community engaged in two-step and multi-step MDLL, and we experimentally tested this theory. Our theory provides a new perspective for predicting ecological dynamics of nature communities and can be applied to guide the rational design of high-efficient microbial systems. Finally, great thanks to my research group. Thanks Professor Wu for kindly guidance of my work, and thanks my colleagues for always supporting me. And specifically, I give special thanks for the ones who helped me finish the experiments with division of labor. In addition, special thanks to my Swiss MSE team to offer me many wonderful scientific suggestions and much support during this special coronavirus time. Thanks for your attention. If you have any questions, please feel free to contact me via email.
All right, and that concludes session 5. Sorry we're a little bit over time, but we'll just keep moving along. So thank you for listening, and I hope you will join us for our final session of day 1. So now I will hand my virtual mic over to my colleague.